side. Let's go to the glutamate side. Peter Kalivas. Welcome.
Hi, everybody. It's always really a lot of fun to come here. So you guys interrupt. I know it's been a long week. Everybody's got to be at least a little bit burned out, but I'll take it slow. If I jump around and we miss something, God, just interrupt, and I'll elaborate, maybe even have a story to tell. So I always hesitate. I used to hesitate anyway to use the word cure, right? It's kind of this megalomaniac thing, like I'm going to cure addiction. And then, did you ever, anybody know who Mark Sanford is? The governor of...
Former governor.
Yeah, the governor of South Carolina. So Mark Sanford, in his last year as governor, told everybody he was hiking the Appalachian Trail to clear his head, get back into the governor's seat. And he actually bought a ticket with taxpayers' money, of course, and flew down to Buenos Aires to visit his girlfriend, his mistress. So he got caught for that, but he was almost done, and so he didn't really end up getting impeached. Well, 2 years later, which is just like this spring, he ran for Congress in actually the district that includes part of Charleston, and won. So if Mark Sanford can get reelected, I can cure addiction.
Good logic.
Okay, so just to set the stage, to give you sort of a perspective on how our lab approaches this, I'm sure you're familiar with this idea that addiction is a spectrum disorder. You've got at one end, you've got social use, which is something we're all familiar with. Regulated relapse would be somebody who maintains a lot of cognitive control over their substance use, and so they can basically decide whether they're going to relapse or not. They have a nice conversation with themselves. They weigh the negative consequences against the positive consequences. And depending on how addicted they are, they'll either relapse or not. A compulsive relapse would be somebody who, as soon as they get the cues to remind them of the drug and the situation that they're going to, the bar where they're going to see their friends, whatever, they basically forget about all the cognitive conversation that they're going to have with themselves, and they just start engaging in drug-seeking behavior. So they start relapsing immediately, or heading towards a relapse. They may be getting in their car and driving to the bar or whatever. So this is a lack of cognitive regulation. This has cognitive regulation, and we may or may not treat somebody like this or try to treat somebody. Depends on how dysregulated the regulation is. And then social use, everybody thinks that's fun, so we don't treat that. And these are the ways we would treat it. Obviously, you need psychosocial interventions for virtually any behavioral disorder. We have some pharmacotherapeutic interventions. You probably heard a bit about those. They're all, in my opinion, they all land more or less right here. Like, they help people who already have a decent amount of cognitive control over their disorder, and they're drifting. They wouldn't be seeking treatment if they didn't have a problem, but they're still able to think clearly, and so the replacement therapy will basically reduce their cravings so that they can cognitively weigh out the pros and cons of relapse and make a better decision, theoretically, not to relapse. What's missing, I think, entirely from our armament of treating addiction, at least pharmacologically, is the ability to convert somebody who's a compulsive relapser into a regulated relapser, where then they're more amenable to the cognitive interventions, the behavioral interventions, and these kinds of nicotine patches, methadone types of replacement therapies. And this is where we've jumped in. And another thing to think about as you think about compulsive relapse going to regulated, how we define addiction is really over here, whether you're addicted to nicotine, heroin, gambling. If you've got a real problem and it's a chronic relapsing disorder and it's disrupting the seeking of biological rewards, you would be categorized as somebody who's addicted, and your compulsive relapsing is part of it. So in theory, anything that's relevant to, say, cocaine addiction that we find here, if it's really part of the conversion process, should work for all addictive disorders. So we would hope that the defining characteristics of compulsive relapse would actually cross between, even out to gambling, but certainly between drugs like heroin, coke, and nicotine, all of which have very different pharmacological actions, as you're well aware. And so that's a theme that I'm going to push, And it's part of the approach that we've taken, especially over the last couple of years, where we've gotten heroin and coke and nicotine self-administration going alongside of the coke self-administration. Okay, so I just start with a brief history that leads to the synaptic plasticity to cure addiction. And I am, in the field of neuroscience, to me, history is not so important. You know, we know almost nothing, as presumably you've all figured out by now. Like, we know very little about the brain. We're all making these little incremental advances, but we are a long way from curing schizophrenia or curing drug addiction. We really are. We don't understand enough about how the brain works. It's like getting in a space shuttle and going to Mars to colonize Mars. I mean, we don't have the technology to do that. We don't have the knowledge to do that. We don't actually have the technology or the knowledge to really cure these disorders right now. So the history is important. I think how I think about it is very self-centered. As I will describe a history of people that influenced me as an example of the types of things you might consider doing as you start developing your career, and the types of people to look out for that can help you as you develop your career. Because ultimately, the people just that are tied with certain findings, it's really the finding that's important. The people come and go, but the finding is what's there, especially at these early nascent stages of knowledge development. Anyway, that's how I've always looked at it. So as a history, what I want to do is just describe to you a couple of people that were very influential in the field of synaptic plasticity and addiction, and also just little stories of how I interacted with them, because I think it's instrumental. So this guy, anybody know who this is? This is Bob Post. Anybody ever hear of Bob Post? Yeah, well, you don't count, Eric. Anybody under 50 ever hear of Bob Post? Okay, yeah. So Bob I think he still has an appointment at the National Institute of Mental Health. You can see this is 1976, and he made a link between cocaine and kindling. So kindling is amygdala kindling, where you stick an electrode in, you stimulate, you induce a seizure, and then you can come back with a subthreshold. You stimulate that a few times, and you come back with a subthreshold level of stimulation, and they'll have a seizure. So they've sensitized. And people were just starting to study the cell biology of what amygdala kindling was. And Bob was studying sensitization to cocaine, so locomotor sensitization. He made this link that maybe LTP, maybe some of these things that were just being characterized back in the 70s might somehow play a role in addictive disorders. He very clearly made this link. Now, he was a psychiatrist and more behaviorally oriented, so he was never really on his own able to pursue what I think is a very seminal insight that he had. He's also a great guy. So this is a guy, and I would encourage you to do this when you've got somebody in your field that's really setting the stage of that field. Seek them out and talk to them. There's nothing people like this like more than for you to come up and know their whole body of work and flatter them and talk to them about their findings and really dig into their work with them. And so Bob is a guy who wrote me letters of recommendation through my whole career. And we're good friends, and it's you want to find mentors like this besides your PhD mentor and your postdoc mentor. There are people out there that really do care and really want to help bring people along, but it's up to you guys to target them and start a conversation with them. Or else the next time you see them, they won't even remember your name, right? You've seen that. That happens all the time. All right, here's another guy. Gordon Mogensen. So who's heard of Gordon Mogensen? Yeah, all right, he's a little harder because he died about 1991. He wasn't that old. He died of pancreatic cancer. And he's the guy, because we'll talk a lot about circuitry, but he's the guy that really put together the idea that there's circuitry regulating motivation, basically cortical and allocortical circuitry, that feeds into the nucleus accumbens, that then feeds out. Now, his focus was not so much on the direct pathway back to the ventral mesencephalon, but was on the output to the pallidum, and in this case, because it was accumbens, output to the ventral pallidum. And he combined electrophysiology with behavior, looking mostly at locomotor activity, but he was the person who really brought that circuit forward. He did this in a review in 1980. Real nice guy, kind of reserved. The interesting story, funny story is that, so with a guy named Israel Hannon and Celeste Napier, we put on a meeting in Chicago. They were both at Loyola in Chicago, called basal forebrain, something. Anyway, it was about the basal forebrain. And we invited Gordon to be our main plenary speaker. So it's about a year before, we're busy putting it together, and his secretary gets hold of us and says, Gordon is dying of pancreatic cancer. And so we go, oh man. So we decide, well, we'll make it in honor. So we did the meeting in honor of Gordon Mogensen. And we got somebody else to do the plenary talk and whatnot. And then about a month before we have the meeting, we get a note from his secretary, there's been a miraculous cure, and he's coming. And we've already printed up all this stuff, honorary stuff. Gordon, I know. She told us that there was like 3 months to go, and he was almost for sure going to be dead. So anyway, he came to the meeting, gave a great talk, and he did die about 6 or 8 months later. But it was, it was very. So stuff happens. It's still, I encourage you to do nice things to people. Sometimes it can get a little uncomfortable, but it's worth it. Okay, so this was another paper. This is Terry Robinson and Jill Becker, and to me, in my mind, they're the first ones to really start thinking a little bit about it. It was all done through dopamine receptors. That's all people thought about back then was dopamine. But how there might be some presynaptic regulation, and they wrote this wonderful review that that set the pace in the field, because at the time, in the 80s, it was almost all behavioral pharmacology. And that's almost all anybody ever did. We were doing a little bit of I was trained as a neurochemist, so we were doing neurochemistry, the HPLC measurements of dopamine and whatnot. But these guys put together this wonderful review that really said, we have to start looking at some of the cell biology of addiction. And then there's, in my life, Jane Stewart. So this is another person who you probably never heard of. She's retired now. She was a behavioral pharmacologist. So like I said, I was trained as a neurochemist, and she knew behavior. And she really, in this thing we put together, she helped me think through this circuitry. This is a modification of Gordon Mogensen's circuitry. And this is something we put together that's based largely on, he didn't give a talk this time, did he? But this is John Williams at OHSU, so a very, very good friend. And he and his mentor, Alan North, we basically just copied all the electrophysiological information from their papers and put it together in this kind of model. But Jane was brilliant and really played a big role in sort of broader conceptualizations of addiction and bringing in things that have nothing to do with dopamine, because behavior is a lot more complex than dopamine. And then there's this guy, who I didn't think he'd be here yet. So, but Eric, when I was in the early 90s, we had just started playing with G proteins, and then all of a sudden, Nestler, all these G protein papers came out. And obviously everybody knows what Eric has done. He's the guy that started cracking open the cell biology of addiction in a big way. And to me, this is what this represents, is finding somebody in your own sort of age cohort or peer group that is 2 steps ahead of you at all times. And you can, so you always have somebody out there that you're working towards. They establish a challenge, basically, in your life. And you can, you work towards that challenge. And to some extent, in the area of cell biology, that's what Eric was. And we were always, keeping one eye on what is Eric doing as we went, followed our own path. But it's really good I think, it was very helpful to me to have people like that in your own peer group. It's different when they're older. You find somebody in your own peer group, people that you really respect, and use them. Obviously, you want to be good friends with them too, and use them to help you think through where you're going and what's up. All right, then you saw this guy. So this is in our own lab. Chris made this breakthrough with glutamate. And this is what really got us started on the idea that glutamate might be important in addiction. And this was an observation that he made with microdialysis in the core of the nucleus accumbens. And so It's a sensitization model. It's not self-administration. We just started doing self-administration here in 1996. And you inject cocaine here, saline injection here, cocaine here. It says extracellular levels of glutamate normalized. And in the animals that showed behavioral sensitization, we got this increase in glutamate. In the animals that did not show behavioral sensitization, or in our saline controls that just got an acute cocaine injection, there was no change in glutamate. So this cued us into the idea that glutamate might be important, but it's important in animals that actually have made some kind of cognitive connection with the drug. So these animals had developed what we'll call a conditioned response, locomotor response, to the daily cocaine injections, the ones that showed this. Those that didn't run around at all did not. And so maybe somehow this glutamate connection, which obviously, if you're looking at the accumbens, is coming in from the cortex, in the cognitive areas of the brain. Maybe that was important in addiction. And Marina Wolf did an awesome job of tying that all together in a 1998 review, and I think really set the playing field for glutamate is important. And at the time, Malinka and Antobanchi were studying LTP, LTD, and she reinvigorated that link based on their work, which was not involving addiction at the time. Anto was, when he came over from John Williams' lab. But she basically, in my opinion, tied that together and got the ball rolling in a big way. So people finally made the connection, had the technology, and went forward with really looking at synaptic plasticity and addiction. And I think it was based on this, to some extent, on this review that Marina wrote. Okay, so now we're at 1998, and this is where my talk will really begin. So the focus was shifting toward drug-induced plasticity in glutamatergic synapses. So finally, it was decided that we really had to find out if plasticity was important. And at this time, people began, not so much the electrophysiologists, but everybody else started using more realistic models of addiction that had more face validity, the self-administration model in particular. And we can talk about that as we go on. I'm not a real big fan of evolving models in these ever increasingly anthropomorphic ways because they're not college students. They're little white rats that live in these very impoverished environments. And so you can anthropomorphize them up to a point where you can't even do realistic reductionist experiments on them anymore because it takes 3 months to generate the animals. Only a subpopulation shows the characteristic. So you would run 100 rats for 3 months and you'd wind up with 12 that you can actually study. And it's not feasible, in my opinion, to do the kinds of reductionist biology that we're going to talk about in some of the more elegant models that have been developed that theoretically are college students who've decided to go snort cocaine rather than go to school or climb a mountain. Just because it's a rat that you've gotten addicted for 3 months and then they're willing to take a more foot shock in order to go get their drug. I think it's an interesting model, but to understand the neurobiology is going to be very difficult using complicated models like that unless you've got hundreds of millions of dollars you can invest in a huge team. So we stick to a simpler model. Okay, so what I'd like to do now is just talk briefly about addiction circuitry and a little optogenetics redux. So basically people have started to use optogenetics to study addiction circuitry. We focus on the relapse event. So we don't focus on reward so much. So I'm going to talk all about relapse. So you're going to see some contradictions perhaps in some of the other talks that you've seen, or what would appear to be contradictions, and a lot of it has to do with where in the model you're studying the neurobiology. So our thought was years ago that if we used a long withdrawal period, we would be getting at not acute changes, not acute withdrawal changes, not allostasis changes. Well, there could be allostasis, but not, what's George? The dark side. Not the dark side stuff that's, the early withdrawal syndrome, but things that are resident that cause people to be vulnerable to relapse months or years later. And so we always have a 2 to 3 hour withdrawal period, usually with extinction training. I'll show you the model in a second. And then all of our measurements are made out there. So we're really interested in relapse circuitry as seen in the reinstatement model. So everybody's familiar with this idea that drugs usurp endogenous circuitry. It's nothing crazy that's going on. And if you think about what it is, very simply framed, you've got habit behaviors. We use habit behaviors 24/7. You know, and how I flip this around is a habit behavior. It's all, I don't think about it. I mean, we develop habit behaviors so we can use our brain and our conscious functioning for new things that might be coming along that could be either really good or threatening to us. You know, snakes do this really well. I mean, this is a lower function. So what happens if your habit gives you an error message? So you're doing something. I go to the refrigerator every night to get an apple. I don't think about it. I can think great thoughts about whatever paper I'm writing or what TV show I'm watching or whatever. That's what's in my consciousness, and I'm habitually go in, get an apple, and I come back out, start eating it. You know, if you ask me when did I take my first bite, or, you know, I don't remember any of that stuff. It's all habit. And I can do all this while up here I'm thinking other things. Okay, then what happens if I go into the refrigerator and there's no apple? You know, well, then that's a big error message. And I enter into a seek and destroy strategy, right? Just start fishing around in the refrigerator trying to find an apple. That involves something like this. So all of these areas start talking to each other, trying to formulate a plan to update that habit so that it will be successful again. And so what I find is that the apples have mysteriously moved to another drawer in the refrigerator, let's say. And so I learned that. I update, and I have now an updated habit, because after I do that once or twice, I'll forget about it again. I'll be thinking great thoughts as I go to the refrigerator and go to the other drawer to get the apple. It'll all be habit. It won't require any consciousness anymore. So this is basically how we get through life in very simple terms, I think. And what our group focuses on is that there's a defect here in this glutamatergic input that's trying to update this habit circuitry in addiction. And so you can't update it as effectively. You can't develop competing strategies, and you can't change the strategies you've got as easily for getting the drug. So in most of what you're going to see when we actually deal with circuitry, this is the projection that we're talking about. It's prelimbic cortex, glutamatergic projection to the core of the nucleus accumbens. And this is the animal model that we use. Very simple. 2-hour sessions a day. Animals will self-administer drugs. And this is the active lever. This is the lever that delivers nothing. And they almost instantaneously, if they're treated right, learn which lever has the drug, which lever does not, and then they develop, in this model, a fairly stable self-administration. We just do a couple weeks, because again, we're interested in them developing habitual behavior to get the drug, which is basically putting them in the box and they start pressing the lever as soon as they get in, because they know that there's drug there. Once they've learned that nicely, which is in our hands not indicated by an escalation in behavior, but by a relative stability of behavior, 3 days of less than 10% fluctuation, then we extinguish the behavior, which basically means that they're now getting saline or nothing. Usually we just let the catheter die out, and they'll press the lever for nothing. And they extinguish that behavior. And you can see they get down here to, after a couple weeks, to an extinction baseline. They're always hitting the active lever more than they're hitting the inactive lever. And to anthropomorphize, this is probably the animal going in and going, you know, maybe. And they'll check. They haven't forgotten that this is where the cocaine was, or the nicotine or the heroin. They haven't forgotten that. What they've done is they have learned to inhibit that pressing behavior if they don't get any drug after a few presses. So it's a very active learning process. I do not have time to talk about it, and maybe somebody else did, but this is a very potentially rich learning process for understanding addiction, because augmenting this inhibitory process could be a way to tackle the problem. I mean, maybe that's part of the defect, is that people can't actually inhibit as well as they could. And so this inhibitory process, we've done a little bit of work studying this, and other people have as well. And I'm happy to talk about it at the end. if anybody wants to, or during lunch. But then we do something to make the animal want the drug enough that they'll jump up and press the lever 100 times for nothing. So they will overcome their inhibitory training and press the lever 100 times even though they're not getting any drug. And there's very anthropomorphic ways you can do this. So we can associate, and this is what we do mostly now, is you can associate a cue and a tone, a light and a tone with each drug delivery here during this training period. They never see that again. And then you don't bring drug back, but when they hit the active lever, a light and a tone go off. And so this cues them that, oh, maybe the drug is available now. They overcome, or it competes and defeats the inhibitory training of extinction, and they press the lever 100 times for nothing. What we've used is foot shock stress. So you do a mild foot shock stress, the lever comes out, nothing's changed, there's no light and tone has come back, and they'll press the lever 100 times for nothing, just in response to being in that stressful, that uncontrollable stress. Or we can give them a little bit of the drug. This would be the equivalent of, you've got this big pile of cocaine and you go, okay, I'm just going to do this one line and we're going home after that. And that would be drug-induced reinstatement, because you can pretty much bet that the person's not going to stop at one line and they'll get a little deeper into the pile. They will actually fully relapse if they start on a drug. It's like a guy in a bar saying, I'm going to have one beer and go home. Okay, so that's the model. And I'll talk about cellular changes here, just before we reinstate, and I'll talk about cellular changes here that are occurring during reinstatement related to the glutamatergic synapses in the nucleus accumbens. Okay, so this chapter had optogenetics in it. This is Rachel Smith right here. So she was the primary author. Mary Kay Lobo, who worked with Eric, is now at the University of Maryland, provided this picture and is a co-author on this Current Opinions in Neurobiology. And this gets to the question that I asked right at the end of William's talk. So if you look here, this is D1, and it's very nicely, it's got the tomato label. It's very nicely labeled, so you can see the direct pathway coming down to the substantia nigra. And if you look in the globus pallidus, there's D2, bright green, and you can see that labeled up. If you go to the ventral pallidum, it's all mixed up. So you've got D1 and D2 innervating the ventral pallidum. So when people say, the ventral pallidum is I think you may know, is innervated selectively by the nucleus accumbens, whereas the globus pallidus is selectively by the more dorsal striatum. So this particular projection is not the clean D1 direct, D2 indirect. At least it doesn't appear to be. And we're doing experiments. Robin's playing a big role in what we're doing. Is we've got some mice from Eric that we can, they're Cree mice, D1 Cree mice, that we can then label, put channel rhodopsin into the D1 neuron, and we can retrogradely label from the ventral mesencephalon or from the thalamus, which have a lot of cells here, and we can determine if it's going to be the D1 or D2 cells by stimulating those neurons with laser light, the channel rhodopsin expressing neurons, and looking to see which of those neurons, D1 or D2, will stimulate the neurons that are projecting out of the ventral pallidum. We're betting that it's going to be all mixed up. And so that's, until those kinds of experiments are done, and William said it right, we just need to do a lot more experiments. I think people need to use caution when they start talking about direct and indirect to the pallidum. This is definitely a direct pathway. That's definitely an indirect pathway. This is kind of a mixed up place that's got D1 and D2 going into it.
I have a question about that anatomy.
Sure.
What's separating the two regions of the palate?
That would be, you can see it kind of fading away here, and it's really actually a line. Yeah. That would be the anterior commissure. Okay. And I'll actually, is William here? Yeah, I actually wanted to show him where the hot spot was in the ventral pallidum for this study. So this is a paper that Mike, so that's Mike Stefanik right there. And I used this purely to get that picture of him. Every time I ask him for a picture, he sends me these really preppy things. And it's just not who he is, so this captures him a little bit better, I think. So he's gone through the brain. It's in this paper, and then a J Neurosci that's coming out, and used halorhodopsin. Excuse me, ARCH-T. Part of it, we started with halo, but then everything else was done in ARCH-T. So we've got if you inhibit this pathway, you inhibit the VTA back to the NA core, or you inhibit this pathway out to the dorsolateral ventral pallidum, you prevent reinstatement. So animals will not reinstate if you inhibit those neurons. If you inhibit the direct pathway, so basically the neurons that are going from here to here, animals reinstate just fine. That is not a problem. The animals do a great job of reinstating when this pathway is knocked down. And there is a GABAergic projection back to the core from the pallidum, and that also did not appear to be important. When we knocked that one down with halorhodopsin, that did not affect reinstatement. So this is the picture, and I think he's already done some work with amygdala input, so it's very clear that the amygdala input here is important as well. And so I think that earlier diagram I showed, where you have all these cortical and allocortical glutamatergic areas that are responsible for providing context and emotion, et cetera, they all input here into the core of the nucleus accumbens. And they're probably all playing some role in the reinstatement event. It might depend, it'll vary depending on how you induce reinstatement. So obviously if you use a cue, which is what we do mostly, the amygdala is going to be incredibly important. If we use a drug like cocaine, it's not so important. So we actually give the drug to cause the reinstatement event. All right, and I just, for your edification, let me just show you this. I put it down here. So this is the part of the ventral pallidum. I don't know if you can see, this is a coronal section, and you can see the anterior commissure. And this is where we did all the fibers were in this very dorsal component. This is GFP from the ARCH-T injection that was made over here in the NA core. That just gives you a sense of, and so you can see the inhibition here. So I wasn't going to go into all this detail, but since we got talking about it. Okay. All right. So, addiction in the tetrapartite synapse. So everybody's heard about the tripartite synapse, right? How many people have heard of the tetrapartite synapse? All right, got a new one. That was a name that, actually the guy that really pushed the tripartite synapse, Rusakov, he's, I don't know where he is, I think he's in Canada, but he's written some really impressive reviews, and they're very synthetic, and he does a lot of mathematical modeling. And so he talked about the tripartite synapse, which is this. You know, everybody is familiar with, I think. You got a presynapse, a postsynapse, and the glia. And together they form a unit that is actually regulating synaptic transmission and synaptic plasticity, as people are finding out more and more. Obviously, pre and post regulate synaptic plasticity, but the role of glia, and we'll talk a bit about that, in synaptic plasticity is becoming more and more apparent. The tetrapartite, the tetra in the whole thing, is the extracellular matrix. Okay, so this is just a blow-up of a synapse. And everybody knows about the proteins in the postsynaptic density. Everybody knows that there's proteins that are tying together the synaptic vesicles in response to calcium binding proteins that are ultimately responsible for stimulus secretion coupling. There's a little scaffold that does that. Well, there is also a signaling scaffold that exists in the extracellular space, not just inside the synaptic cleft, but outside the synaptic cleft as well. It's very tightly regulated, and it's tied to some extent to synaptic transmission. And it is necessary to manipulate this scaffold if you're going to induce any kind of long-term potentiation or depression in synapses. And so we'll go over some of that. And we found that this is also potentially involved in the synaptic plasticity associated with addiction. With reinstatement, I should say. Okay, so this is just a cartoon that lays out things, and I'll come back to this cartoon as we build a model of the synapse in addiction. Yeah.
Just a question about the extracellular matrix of the brain. I know in like muscle and stuff, it's collagen, fibroblasts, and everything. What is it in?
They're there. There's laminin, fibronectin. I mean, I actually don't, I don't have the list in my head, but there's a bunch of them. The two we've approached it not so much from the proteins themselves, but from the enzymes that remodel the extracellular matrix. So the matrix matrix metalloproteases, right? MMPs. Sorry, and that's we've focused in on 2 and 9. Those two, they're the gelatinases. So laminin and fibronectin, for example, are their substrates. And I'll show you how we kind of got tuned into those in a minute, probably after lunch, actually. Okay, so this should all be pretty familiar. You have AMPA and NMDA receptors that sit inside the synapse and do the signaling. You also have these extra, we'll talk about this little calcium business in a minute, but you have these extrasynaptic, or what are thought to be predominantly extrasynaptic metabotropic glutamate receptors, the group 1s, the 1s and 5s, and then the NMDA receptors that contain the GluN2B subunit are thought to be more extrasynaptic than inside the synapse. They're a little bit everywhere, but most people think that that's the general distribution. You have presynaptic metabotropic glutamate receptors, so these are, for purposes of what we're going to talk about, just classic autoreceptors. So it's GI-coupled inhibition of stimulus secretion coupling through the mGluR2, mGluR3s can also do it. These are GQ-coupled. So that's why you've got the calcium there, because they can release calcium from the endoplasmic reticulum. And for the tri, what we're going to focus on is glutamate transport. So glutamate transporters, and again, this is because it's hard to visualize this kind of stuff, but certainly in the cerebellum and the hippocampus, mature synapses have a patterned expression of highest density GLT1 right at the synaptic cleft. So they're on the glia. It's almost exclusively glia, this particular transporter. And they sit right at the synaptic cleft. So the idea is, then you've got the matrix metalloproteases. Okay, so they live out here, and they're the two that we study. MMP2s are constitutive, and they do get affected, at least by cocaine. The MMP9s are inducible, and those are definitely affected during a relapse situation, and I'll talk about that. And maybe I don't put I do. So you have glutamate release, and this is kind of what you'd expect. Let me show that again. I've only got a few animations like this. Got to see them a couple times. So it's you release, you go to those receptors, and then most of the glutamate gets eliminated through the uptake system, and you get a little bit coming out here. So if you have a lot of activity here, you're going to get a little autoreceptor stimulation to decrease release probability, and you'll start getting stimulation of these extrasynaptic receptors. Cases are being made, of course, that you've probably heard of mGluR5 induced LTD, and there's a lot of GluN2B induced LTD and LTP that people have studied. So this spillover, as we call it, this glutamate spillover might be very important in as much as it's stimulating these to produce postsynaptic plasticity. We'll talk about that. All right. So what do addictive drugs do? This is my craving slide. Is it working? Okay, maybe not. What do addictive drugs do to disrupt this glutamate homeostasis? And by that, we're referring a lot to the release of glutamate and the fact that glutamate gets eliminated largely by those glutamate transporters, maintaining the fidelity of excitatory synaptic communication. So you don't have all this spillover, not a lot of crosstalk between synapses. It's point-to-point communication, which is probably very important. If I've got 20 thoughts going on at once, each is going to have a little bit different neuronal ensemble that it's communicating. If you start fleshing between them, it seems to me it would be confusing. And possibly, as you'll see, that's what's happening in addiction. Okay, so let's just take a look at GLT1 and it's what it's doing in these different models of different drugs. So you've got cocaine. Cocaine reduces GLT-1 levels, and this is, if you look at glutamate uptake, you see the exact same thing. Glutamate uptake's way down. It's very dramatic, actually. Nicotine, same thing. The protein effect was not so great, which was what was in this paper, but we've recently done an uptake assay, and it's pretty strikingly reduced, glutamate uptake. This would be sodium-dependent uptake, those of you that are familiar with these uptake assays. So it's the GLT-1 type of transporter. Alcohol, same thing. You get a reduction in GLT-1, pretty dramatic. These are all in the nucleus accumbens. Some isolated the core from the rest of it. And here's heroin. Very nice reduction. This is a paper that's in submission right now. And so, and the uptake is also reduced. So all of these drugs share in common a reduction in GLT-1 in the nucleus accumbens. Okay, so what's that going to do? Well, I've already more or less described what it's going to do. Here's the normal situation. You've got glutamate coming out, disappearing, little talk here extrasynaptically, but not too much. If you downregulate this, you would expect something like this to occur, right? You get this overflow into the extrasynaptic space, and you would start getting stimulation of these extrasynaptic receptors. Okay, so is that happening? Well, the easiest way to do that, and historically in particular, is you just jam a dialysis probe in. Now, we all know that that's a huge thing. It's not going to be sitting inside of any synapse. It's only going to pick up the stuff that flows out of a synapse, not the stuff inside the synapse. And so it has to make it out, make it through the gauntlet of GLT-1 to get to the dialysis probe. Or you can use more elegantly a biosensor. So it's basically voltammetry, only it uses, it's got an enzyme that degrades glutamate and allows it to be oxidized by the current. And so it's essentially like measured dopamine with a voltammetric electrode. You can get better time resolution, but it's still way too big. Even though the small ones are down to 10 micron diameters, it's still way too big to sit inside a synapse. So the only thing it's going to pick up is overflow. It comes out of the synapse. All right, so this is an observation. Now we're all in, everything's in the self-administration model, but this is very similar to what Chris found with sensitization, except for virtually all the animals reinstate. And we used to think this was incredibly important when we were just working with cocaine, this reduction in basal glutamate. So there's basal levels of glutamate in a control animal, and the coke animals had less. And this became, the little lure that we followed for like a decade, thinking that this was really where it was at, and we were going to discover what these basal changes were all about. And we got in trouble from reviewers who didn't like our ideas about it. I mean, it was a very exciting time. Turned out it's probably not so important in addiction. If it is, it's very selective for cocaine addiction, this particular reduction. What you also see is whether you give a cue, a stress, or inject the drug, you get this big release of glutamate here in the coke animals, the ones that have been trained and extinguished from cocaine self-administration, versus the yolk saline, where you don't get, essentially nothing happens unless you give a really high dose of cocaine, then you can bump a little glutamate. So this is all synaptic. So if we give TTX, that doesn't happen. So we put TTX through the dialysis probe in the accumbens, inhibits stimulus secretion coupling, and you do not get this big release. If we inhibit the prefrontal cortex, so back in 2003, you do this by putting baclofen and muscimol in the prefrontal cortex, GABA agonists, you inhibit neuronal activity, you prevent that. So that does not happen. This has nothing to do, as near as we can tell, with synaptic glutamate. At least if you put in TTX, let's just take the control levels, they stay the same, rock steady.
What if you electrically stimulate prelimbic cortex?
If you stimulate hard enough, you'll get a bump. But it's yeah, you have to.
Physiological probably.
Yeah, nobody really did those experiments with this particular pathway, but other pathways that are glutamatergic, people have shown that with pretty aggressive stimulation, you can get overflow. So it's, I'm not going to show any of this data, but we've actually set up an assay to measure overflow where we use NMDA currents, which is, as we increase stimulation frequency, the NMDA current, the half-life of decay gets longer. and we can show that in a slice taken from an addicted animal, you get a curve like this in a control animal. There would be one stimulus, and if you buzz them 4 times, you know, over the course 4 times in a row, every 10 milliseconds, I think, over the course of a minute. I can't actually remember the parameters on this experiment. Sorry. You'd get something like this, where you start the decay constant of the NMDA current gets gradually bigger. In an addicted animal, it looks something like that. So we've at that level where you're actually recording the near adjacent NMDA receptors, you can definitely demonstrate that even with a single stimulus parameter, you're probably getting some overflow. The question is, you know, what is this big thing that we're seeing here? So this turns out to be cysteine glutamate exchange mediated, and there's a real problem with that in cocaine addicted animals. It does not appear to exist when they're addicted to nicotine or when they're addicted to heroin. And so that we became big fans of what's causing this, which, as I described, good money would be on downregulation of glutamate uptake, because that is what's limiting that synaptic release from getting out into the extracellular space. Okay, so just to go through it, this is Q-induced relapse in nicotine. If you actually give a nicotine injection, you get a much bigger one. Anyway, there you can see the increase in glutamate release. This is a biosensor done in an alcohol self-administration protocol. And this is a guy named Justin Gass did this study when he was working with Foster Olive. And then these are heroin animals. And you can see this big release that you get when you give an injection of heroin and the animals relapse. So all of them produce this synaptically dependent release of glutamate. And they all have a downregulation of GLT1. All right, so what else do you find? Again, at 3 weeks of withdrawal, usually in an extinction paradigm. So what else is going on besides these 2 things? Downregulation of GLT1 and then this overflow during relapse. But really, just basal state, downregulation of GLT1. So these are things that are all shared by nicotine, heroin, and cocaine. So we're getting beyond what's the binding site that's initiating reward. We're getting beyond reward circuitry in general, and we're really dealing with relapse circuitry. Okay, so there's this Scott Bowers. He was a graduate student, went on to do better things. This was his paper as a graduate student, and then he carried on most of this. This was not his paper here. And basically showed that there's a reduced mGluR2 signaling. Other people have seen this just by looking at mGluR2 receptor surface expression and things like that. But what he found is that there's this G protein binding protein called AGS3. Activator of G protein signaling 3 is what it's called. It actually inhibits GI signaling. So it competes with beta gamma. So you have let's say mGluR2 glutamate binds. The alpha subunit and the beta gamma separate off and go do their signaling thing. This AGS3 binds to the alpha subunit and prevents the heterotrimer from reforming. So it basically downgrades signaling through GI coupled receptors. And that's upregulated in all of these drugs in the nucleus accumbens after self-administration of all these drugs. And the other one is there's upregulated gluN2B containing NMDA receptors. So you can see it in coke animals, heroin animals, and nicotine animals. What things have people studied a lot that do not cross between drugs? Okay, so there's an increase in AMPA receptors. So there's been a big story built on the cocaine world. We've helped contribute to this. When we're going for what are the adaptations that are critical to addiction, not just cocaine or nicotine or heroin, but to addiction in general, what you find is that cocaine and nicotine produce a very nice increase in AMPA receptors in the nucleus accumbens, measured either with protein, looking at surface expression, or doing an AMPA to NMDA receptor or current ratio. And heroin does the exact opposite. So animals withdrawn from heroin have a lower AMPA to NMDA ratio, and The other part is the spines. So you guys are familiar. I'll show a little cartoon here in a sec. When you go into LTD, spines get bigger and you stuff in more AMPA receptors, right? Into LTP, and you go into LTD, and the opposite happens. And synapses float somewhere in between there, depending on, that's sort of grading the synaptic strength of the synapse. So the part and parcel to looking at AMPA receptors is you look at dendritic spine density and or spine head diameter. And it's the exact same thing. That's cool for cocaine because you get a very nice increase in density and spine head diameter. Same thing with nicotine. The exact opposite happens with heroin. And so these are all, again, self-administration paradigms in withdrawal. So these things they're interesting phenomenon to study, and you can work out the molecular biology of how it's happening, and I think it's very important to do. But they may not be tied in with this shared trait that we call addiction, that's shared by heroin, cocaine, and nicotine. And here's this was our sad moment. So we had this one great paper that got us going down this road, like, this is it. We've got we've nailed it. All we got to do is get drugs that work on this. And we had some really good, we had a drug that worked on it and everything, but it turned out it also, as I'll describe, upregulated GLT1 too. And it's really the GLT1 that is important. But anyway, neither nicotine or heroin really do this. And then finally, I'll show you after lunch the matrix metalloprotease story. And the basal change in MMP2 appears, we've just got, this is all an evolving thing, but we've just done the first heroin animals, and it does not look like it's up in the heroin animals. But the relapse increase in MMP9 is, and we'll get to the relapse events here in just a second. Okay, so basically, there's a lot that's being studied right now that is not shared by these addictive drugs. And there are a few things that are. And those things are, and there's probably others that people haven't found out yet, so this downregulation in GLT1, this increase in NR2Bs, and this decreased signaling through mGluR2 receptors. So what's that going to mean? You'll have a decreased ability to put the brakes on release probability. So now when the prefrontal cortex goes, hey, I really like that cue, that means drug's available, and it sends a signal down to the nucleus accumbens to engage the behavior, the appropriate adaptive, well-learned behavior, it won't have an automatic shutoff, or at least not as effective for one. And you'll have this downregulation of GLT1, so glutamate will be flowing out all over the place, and you have an upregulated NMDA, gluN2B, NMDA system.
Question real quick. So is that, you have those in the extrasynaptic space, but is it also at the synapse, the change in the NR2?
The gluN2B? Again, it's controversially right. And the reason it's controversial is because the methods for determining that are fuzzy. And so you can activate this, give MK801 and do a small activation where you think it's all staying in the synapse, and MK801 will block all these, then in theory everything else you're measuring when you do the next one is extrasynaptic. So that's one way to do it. Memantine is thought to selectively inhibit these. We could never demonstrate that. There's a paper out there, and we really tried to do that. And we just, it's probably in there, it's just not good enough. And it's just like the MK801 experiment. Well, how hard are you actually going to stimulate? How much will actually slip out, and which NMDA receptors will actually be open? It's good, but there's that user difference potentially. And we know from the experiment that I described in response to that question that really just one or two stims, and you will get a little bit of overflow that at least we can pick up by this gradually increasing NMD, decay constant in the NMDA current.
One more question. Just curious, all those changes you mentioned, GLT transporter and GluR2 and N2, are they have a difference in the response of a changing? Like some of them only happen earlier, some of them happen later?
It's a big weakness in the studies that we do. We look at basically one time point. We just withdraw them out, And then we look at that time point. Exactly, we've on occasion done some of the early time points. So for example, the AGS3 increase, not so strong in early withdrawal, but it comes on by about a week. And that appears to be, there's other proteins that do stuff like that as well. Like this, the well-known, at least in the glutamate addiction people, where you have the GLUR2 substitution for GLUR1. That appears to take place over time in withdrawal. And there are a few other things that are like that. But this one, I do not know the answer to that. And this is, I think, I mean, it actually, we should study it because we think this is probably very key to how we're going to approach the problem of addiction pharmacotherapeutically. And this, the Coke data, which Dong did, it's there at 3 days. There at 24 hours, he had these silent synapses that he saw.
So in the data you show the decrease about 50% around the transporter, the glutamate transporter. Is that how long you have that data?
That's 3 weeks of withdrawal. Yeah, all of those treatment paradigms that I described. The alcohol one, we didn't do that work, and so I don't know the exact withdrawal time, but it was maybe a week of withdrawal probably. That's more typical in the literature where people do like a week withdrawal period. Okay. We can break here, or I can do another. There's 9 chapters, so. What we're going to do next is talk about. So let me just show you. So just set the stage while you go out for lunch. This is what we've been talking about. So what are the enduring? What is enduring in the addictive state? So 3 weeks of withdrawal. What's sitting there inside the brain? What I want to describe to you next, which we think we've come to really focus on over the last couple of years, is what happens during the first 15 minutes of relapse. What is the cell biology of change? And it's quite dramatic, as you'll see, that's going on right here. And maybe this is the stuff we want to target. So you can do stuff to help decrease the vulnerability, but what if you could do stuff to prevent the cell biology of the relapse event itself? And how effective would that be in helping a person regulate their vulnerability to relapse? Okay, so we'll leave it there, and I'll see you after lunch.